Good evening, doctors, and a very good evening to our speakers, Dr. Rahul Pandit, Dr. Amit Gupta, and Dr. Ravi Doshi, on our uh, third series of webinar on Immunocin Alpha, that is Thymosin Alpha 1. And with this, I'm handing over the session to our president, Dr. Devesh Das.
Thank you very much, Samrat. On behalf of Gufic Bioscience Limited, Mumbai, myself, Dr. Devesh Das, President, Domestic Formulation, India and Nepal, do welcome all of you for this webinar. Now, those who are attending our webinar, they are aware of that there is a series webinar we are conducting, and this is the third series of this webinar. Earlier, we have taken different speakers and different case study presenters. This time, we have with us Dr. Rahul Pandit from Mumbai, Dr. Adarsh Shetty from Mumbai, Dr. Amit Gupta from Kolkata, Dr. Rovi Dosi from Indore, and Dr. Navneet from Delhi as our panelist. Now, this webinar is telecasted directly through YouTube and Zoom also. So doctors of Nepal and India, they are watching it. So I welcome all of you. I also welcome our Executive Director, Mr. Pranab Choksi, our CMD, Mr. Jayesh Choksi, and all Gufficians across the country. Now, if I give a small background to our new joinee delegates, thymosin alpha as a molecule, which was being approved around 38 years back by FDA and also DCGI has approved. It is actually now a repurposed drug, and few months back in China, it has been tested. The trial was there for more than 2,000 patients on COVID-19, and a beautiful and very successful results were there. With this background, we as a very responsible pharmaceutical company, under this COVID-19 pandemic, we thought we should bring it to India. And we are the first pharmaceutical company who has brought it to India. And we have applied for DCGI approval for COVID-19. The DCGI has permitted us for phase 2 trial in 6 centers, which is going on. Before that, we have started investigator-initiated trial in 3 centers of Bombay. One is Nanavati Hospital, another is Fortis Mulund, and also we have initiated the and the effect of thymosin alpha on T cells, that has been also taken by our respected Dr. Om Srivastav sir. And we have seen the case studies across the country. Dr. Vimal Ranka from Ahmedabad, Dr. Chinmay Godbole from Mumbai, Dr. Ankur Gupta from Indore has taken the case studies on thymosin alpha. And I'm very happy to share with you that in this last 1.5 months time, we have the data of more than 1,000 patients who are being used mild, moderate, and severe cases of COVID-19 across the country, and there are 400 prescribers who have given us testimonials and their feedback after getting the good result. The Immunocin alpha, that is Thymosin alpha, is now available in most of the corporate hospitals and big hospitals. I'll name some of the hospitals like Nanavati Hospital, Josluk Hospital, KEM Hospital, Max Group, Fortis, Care Group, NH, Peerless Hospital, Kolkata, Medica, BL Kapoor, Delhi. So the response which we are getting, it is really encouraging. So without wasting any further time, I'd like to inform you the flow of today's program. So today also we are talking regarding thymosin alpha, a boon for COVID-19 patients, which will be taken by Dr. Adarsh Shetty, Mumbai. And then we'll be talking on the investigation initiated trial, which we have conducted for the first time in the country, that is Fortis Hospital, Mulund, Mumbai. So we will be talking on which protocol we have followed and what is the bullet points of outcome, which will be taken by Dr. Rahul Pandit from Fortis Mulund. He was the chief investigator in that entire trial. Although the entire trial report and very, very in-depth report will be publishing, and we'll be showing it to you with due course of time. And we have 2 case studies today, which will be taken by Dr. Amit Gupta and Dr. Ravi Doshi. And this will be our program flow. And the entire program will be coordinated by Dr. Navneet Vadva. So very quickly, I will give a brief introduction of our all eminent speaker today, Dr. Rahul Pandit, who is the Director, Intensive Care, Fortis Hospital, Mulund, Mumbai. He is the Vice President of ISCCM and Chairman, ISCCM, Mumbai Chapter, and member of COVID-19 Task Force of Maharashtra Government, had many publications nationally and internationally. And he is also our chief investigator for our first trial. Dr. Amit Gupta, our one of the eminent speaker who will be presenting the case. He's from Kolkata, and he's associated with many hospitals of Kolkata. Especially, I will mention Bellevue Clinic, Nightingale, Kothari, INK, GD, Woodlands. He's a MD, and basically he's from Kolkata. So he will be taking his case today. Dr. Adarsh Shetty, who will be taking you through thymosin alpha, a boon for COVID-19 patients, the major topic of today. He is MD from Banaras Hindi University and having more than 15 years exposure in medical marketing. And he is the person who is instrumental to get the DCGI permission and DCGI interaction on behalf of GUFIC and on behalf of the entire organization. So he will take you through the product. And as I have told you, that we have Dr. Ravi Doshi from Indore, who is the professor in HOD, Department of Respiratory Disease, Sir Aurobindo Medical College and Postgraduate Institute, Indore. And he has a lot of achievements and lot of accreditation publications also. And our coordinator, Dr. Navneet. Dr. Navneet is a MD from KMC Manipal. He is having his own venture, that is AUW Global, and he's the external medical consultant of GUFIC Biosciences. As you know, today, in fact, worldwide, yesterday's data was around 3.60 crore patients have been detected with COVID-19. 10 lakh deaths were there. In India, around 70 lakh patients were there detected, and 1 lakh deaths are there. And daily average of 70,000 patients we are getting. So we, as GUFIC, as a pharmaceutical company, wanted to give you some weapon to tackle this pandemic and to strengthen your hand so that the patients get recovery and patients get benefited. With this, I hand over the session to Dr. Navneet. Dr. Navneet, please.
Yeah. Thank you so much, Dr. Devesh. It's my privilege and honor to be part of this forum. Uh, I think it's very, very unique uh, uh, timing in terms of basically, it's my honor and privilege to be part of this forum. I think the voice is
It's audible.
okay. The voice is echoing actually at my end. Uh, it's my privilege and honor to be part of this forum. I think the timing is so unique that as the as the cases are growing, as the cases are growing continuously, uh, what is required at this juncture is the evidence-based perspectives, how things can translate into better management of COVID. Yeah. And with this, I'd like to request Dr. Adarsh Shetty to take us through the basic perspectives of the molecule and where it fits well. And as the discussion progresses, we will invite the clinicians to share their own perspectives as well.
Yeah, thank you, Dr. Wadhwa, for the kind introduction, and thank you, Dr. Das, for the introduction. I want to thank everyone for attending the webinar. I'm audible, right? And my slide is visible, right?
Dr. Shetty, can you be a little louder?
Yeah. Uh, Thank you, everyone, for attending this webinar, and good evening, everyone. So we are bringing this molecule called Thymosin alpha 1, a boon for the COVID-19 patients. This is the peptide with the 28 amino acid, which is given subcutaneously. So outline the presentation will be, I'll be talking mostly on the T cell role in the COVID and what is the role of CD4, CD8, NK cells and T regulatory cells in the mild versus severe COVID. Then I will touch upon the basics of innate and adaptive immunity, just to 2 to 3 minutes. Then we'll move on to the product, thymosin alpha 1. Then we'll move on to the mechanism of action, pharmacokinetics, pharmacodynamics, and what is its role in coronavirus, both SARS and SARS-CoV-2. And we'll also touch upon how the thymosin alpha 1 reduces cytokine storm, since everyone knows that cytokine storm is the one which causes the mortality. Then we'll touch upon the T regulatory cells, which is also called the regulator of regulators, which are also called the regulator of regulators because this has not been touched upon by many things, because this is the one which causes the immune homeostasis and tissue homeostasis. This T regulatory cells, also called Treg, which is decreased in severe COVID. And because of this, because everything is moved to the lungs, and there's a decrease in T regulatory cells. And we'll touch upon the direct viral actions of T alpha 1 and the trials. If you have some time, we can touch upon the trials. Right now, 3 trials already completed and published, and 2 trials are ongoing in US, and 1 trial which has been published in the prophylactic also. So these are the various publications which has been published till now, and you can clearly see that the as and when the COVID severity increases, there is a depletion of T cells. Mostly the CD4 and CD8 count decreases, and the neutrophil to lymphocyte ratio, which is the main prognostic marker, as and when the NLR ratio increases, the patient moves to the severity mode. It is said that even the increase in one number of NLR1, which will increase the mortality rate by 5 to 10%. And it has been shown that the reappearance of effector T cells is associated with the recovery of COVID-19. So there are various publications which is going on T cells and the COVID-19. So coming to the T cell response, T cells plays the fundamental role in COVID-19. It is seen that the T cell response, which is good in the mild cases, they don't move to the severe cases. And the CD8 cells, which the MHC class 1, which will recognize the CD8 T cells, which will directly kill the viruses. So it's a dysfunction of the T cell response, which is the main pathology. So to better understand the T cell response in COVID-19, the two questions one should ask is, what is the contribution of T cells for the initial virus control, and how is the T memory cells which has been established? Right now, the vaccines is going on, mostly the vaccine which produces the humoral immunity, but right now the talk is the vaccine should also increase the T cell response. And the T memory cells, if you check the previously who are infected with the coronavirus, they are only getting milder symptoms. So the T memory cell, which is main important factor which one has to look into. So this is the main thing where the CD8 cells, which directly kills the virus infected cells through MHC class 1, and CD4 T cells, which will kill, which will activate the macrophages, which is the part of innate immunity. So MHC class 2, which will be recognized because MHC class 1 and class 2, which are decreased both in this one, severe diseases. And the Th2 response, which is stimulated, which will help in the production of antibodies, B cell activation. So this is the proposed model of CD4 T cell responses. You can clearly see that in the milder cases, there is a T cell response is present. So when the viral load is less, when the T cell response is good, then the patient will recover easily. As and when if you check the severe disease, where the initial T cell response is decreased, but the later stage, the T cell, when the viral load keeps on increasing, then the more number of T cells are activated, which will cause a cytokine storm. So it is a T cell response, initial T cell response, which will tackle the disease very well. And some of them will go to the T memory cells also. So there are some markers called the PD1 and TIM3 markers, which are the exhaustion markers. If you can check in the severe disease, where definitely there is a lymphocytopenia, in that the PD1 and TIM3 markers are increased. These are the exhaustion markers because of the Th1 hyper response. The cytokines are produced very much, and the T cells are exhausted. So this is thymosin alpha 1 is the one drug which which prevents this exhaustion, and which nullifies the action of this PD1 and TIM3, so that the lymphocytopenia is corrected. Coming to the CD8, is the same thing, where the lesser activated in the severe disease because of the exhaustion. Again, the markers are PD1 and TIM3 markers, and which will terminately differentiate the T cells, and which will be exhausted in the later end. So the patients with COVID-19 with severe pneumonia reportedly have substantially lower lymphocyte count and a higher plasma concentration of number of inflammatory cytokines such as interleukin 6 and TNF alpha. Again, coming to the NK cell functions, NK cells are decreased again in the severe disease. NK cells, as you all know, that decreases the, it attacks the virus infected cells, and it will decrease the viral count or the viral load. And this is the one mechanism why the people move to the severe disease, because the risk factors are listed here, where mostly the comorbid conditions and the chronic kidney disease, where the T cell response is very less. Mostly because in these comorbid conditions, there is a decreased expression. Some of the theories which are postulated are there's a decreased response of MHC class 1 and class 2, and there is something called PRR, is also called pathogen recognition receptors, which will differentiate self versus non-self, which are impaired in the comorbid conditions. So these are the PAMP, also called the pathogen associated molecular patterns from the virus, this one, DNA or the membrane, which are recognized by the PRR, which will again activate the innate responses. And it is said that the coronavirus, which will encode a protein, which will mask this PRR. So this is one of the things which causes initial decrease in T cell response, and the patient will move to the severe disease. And coming to the typically how the T cell response happens is usually it will take 7 to 10 days to prime the dendritic cells and expand the T cell response. So if the patient is mild, you'll definitely recover within 7 to 10 days. That means the T cell response has happened and the patient has recovered. And if you check the severe disease, if you check the severe disease, the T cell response contributes to the SARS-CoV viral response depending on the severe category. In the severe category, what happens is the viral load is increased and the initial T cell response doesn't happen. So when the viral load is increased, then automatically there are billions of viruses. Then the T cell response, Th1 response happens. Then it causes the cytokine storm and the exhaustion of T cells. So this is the main cause of mortality. So it is always the T cell response which is very important in the initial phase. Again, to collaborate with that, in this article, we can see that the milder disease as a T cell response initially, and the severe disease as the T cell response high, because it collaborates with the viral load. So once the viral load is more, the T cell response is more. Now it is essential to understand the therapeutic intervention and how to time the T cell response. It is critical to determine the balance at an early stage of the infection. So in the presence of robust memory T cell is very important. So it has been checked in the blood of the donor, and it has been found that in the milder cases there is a memory T cell response and the T cell response. Now you may ask how to check the T cell response. The T cells, it's almost 10,000 times bigger than the B cells. Till now, there is no, now the diagnostic tools are coming up, but till now there are no diagnostic tools to check the T cell response. So it's because of the bigger size of the T cells. Understanding the roles of different subsets of T cells in protection, which is very crucial in the prevention and also in the treatment of COVID-19. Again, it's the same thing where the viral load is decreased, the T cell response, if it occurs at the initial days, then the virus is cleared by the T cells, that's the Th1 cells. So if this 3 to 4 days are missed, then the viral load will multiply like anything. Then the T cell response happens. Then a lot of T cells are activated simultaneously, which will lead to the cytokine storm and which will lead to the mortality. You can check in this figure where the mapping is seen of CD4 and CD8 cells and in the resolution phase in both mild and severe disease. Again, coming to the this one, in the previous, if the patient is previously infected with coronavirus, the T cell response has happened, then you can check that cases that those patients will have only milder symptoms. And also, if you check the SARS patient, which has happened in 2002 or 2003, even after 17 years, T memory cells were present. So the T memory cells is one of the important things, and even in the vaccination, in addition to the humoral response, even the T cell response is very important. And the recent T cell studies support that the vaccine modality is designed to induce both cellular and humoral, not only the humoral, because the antibodies in the this one vaccine, this COVID, which will stay only for 2 months to 6 months. So definitely it's the T cell which is more important, and the response of the T cells. And this is one of the French study where they have found that the 6 out of every 10 patients there is a T cell response, and there was no antibody in their body, even though they are exposed to SARS-CoV-2. So which tells the importance of the T cells, because the T cells will be clearing the virus initially, and if you miss the initial response, then the severity will happen. And it has been seen that the lymphocytopenia is a less documented finding in children. So which collaborates with the T cell response and the good immunity and innate and adaptive immunity in children. And in one more study called the Swedish study, it has been found that the 200 people found strong T cell responses in most individuals, had mild or no symptoms, but no detectable antibodies. Again, which talks about the T cell response. Coming to the laboratory markers, the T cell response is very difficult to find, because you can check in the figure that the blue color is the B cell and the red color is the coronavirus. And if you come to the blue figure, you can check that the blue one is the T cell and the small red one is the coronavirus. You can check that it is T cell is almost 10,000 times bigger than the B cells. So laboratories T cell response is very difficult, and very major laboratories you can find this T cell response. And this thymosin alpha 1 was used in the SARS-CoV in the SARS 2002 infection, where it was found for the effective controlling and for preventing the spread of SARS-CoV, SARS-1. So this is documented in one of the journal called Journal of Medical Virology. It has been found, it has been used in China in 2002 outbreak. And it is said that because of this drug, it decreases the spread of SARS. Even though it's almost similar to 80%, genetic is almost similar to 80% of SARS-CoV-2, the spread was limited. And it has been proposed that because of this drug, it has decreased the spread. Coming to the same thing, where because of the decreased peripheral T cells associated with disease severity and inflammation is now well documented, and several studies have shown that the CD4 and CD8 T cells, which is the main friend, which are exhausted in the later severe disease stage, and which has been found in the markers like PD1 and TIM3 markers. And you can see the figure that peripheral blood T cells keeps on decreasing as the disease severity increases. And you can check that the viral load increases, inflammation increases, and disease severity increases as time passes. And this is the lymphocyte, neutrophil lymphocyte ratio, NLR ratio. It is proposed that is one of the prognostic markers. It is said that it's one of the chief prognostic markers, as neutrophil to lymphocyte ratio increases to greater than 4 to 5, more or less during the time of hospitalization. It is said that those people are prone to develop severe symptoms. And lower CD4 and CD8 NK cells, and even including B cells, are seen in the severe COVID-19. And PD1, TIM3 markers are the exhaustion markers because of the excessive stimulation of TH1. The lymphocytes are exhausted, and the thymosin alpha 1, which prevents this exhaustion. I will talk about these studies in the later in the clinical trial stages. This is one of the study where it is found that while the appearance of the T cells, effector T cells, that is active T cells, which is associated with the recovery of COVID-19. This one study which was conducted in the Biomedicine Journal, which shown that the activation and expansion of innate adaptive immunity play a major role in COVID-19. That is well known. And it is found that when the exhaustion happened, when the improvement in the exhaustion took place, it was seen along with the recovery of the patient. And this is also which is published in the Wuhan, China, where the suppressed T cell mediated immunity in patients with COVID-19, which was seen that the severity of COVID-19 is getting worse as and when the T lymphocyte came down. And the patient died in the hospital, had more of a decreased CD4 and CD8 count and B cell count, which was found in the severe patients. The conclusion was it is well known now that the decrease in T lymphocyte, which is seen in the severe COVID-19 because of excessive stimulation. So this NLR may be the important predictor markers, prognostic marker for initial hospital admission. So we'll talk about just scroll about the this one, basics of immunology. Usually the antigen presenting cells will present the antigen to the T cells, then the T cells to the interleukin 2, which will activate the cytotoxic T cells, that is the CMI, then the T helper cell 2, which will activate the B cells. Usually in the vaccine, it has to activate the TH2 also. TH2 is going to activate the B cell, which in turn is going to produce the immunoglobulins. So the interaction between the dendritic cells and the helper cells, which is the link between the innate and adaptive immunity. So just to scroll upon firstly, coming to this virus, the B cell is this is MHC class 1. MHC class 1 is going to attach it to the CD8, and MHC class 2 is going to attach to the CD4 cells. So B cell is going to present antigen to the T cells. In the T cells, the cytokines comes, the cytokines will, and the plasma cells will produce the antibody, and it is going to attack the viruses. Usually the MHC class 1 and MHC class 2 are decreased in the severe diseases. And the CD8 T cells, which has a direct killing effect, which will recognize the through MHC class 1, and CD4 T cells, which will stimulate, will activate the macrophage, which is a part of innate immunity, and TH2 is stimulated, which produces the antibody production. So all the vaccines should activate the TH2 and to produce antibody. The problem is the T cells doesn't have the antigen receptors on the surface, and the B cells have. That's the reason antigen presenting cells is present to present those antigen to the T cells. So these are the various classes of APCs. The DIRT is going to attach to the MHC class 1, and MHC class 1 is going to present. One is going to present to the CD8, and MHC class 2 is presented to the CD4. So this is the MHC class 1, which is going to present it to the CD8 cells, which will recognize, and the virus infected cells are going to get killed. And the MHC class 1 is class 2 is going to attach it to the CD4. So again, the part of this one where the antigens is recognized, and it will present it to the MSC is going to present to CD4. CD4 is getting activated, and which is going to release the cytokines. This is the antibody production, where it's already discussed, where that the Th2 which will help to getting the antibodies. So this is the cytotoxic T cells which will release perforins and enzymes, which is going to kill the virus infected cells. So thymosin alpha 1 is going to increase both CD8 and CD4, and which is going to prevent exhaustion also. I'll talk about it later, coming to the mechanism of action. So thymosin alpha 1 is a drug which is the hormone which is synthetically produced from the thymus gland. So this is a 28 amino acid peptide which is produced by the thymus gland. So coming to the mechanism of action, this drug has a unique mechanism of action, where it has a different mechanism of action depending on the body conditions. Coming to it improves the Th1 response in the mild and prophylactic cases, and it will increase the NK cell activity, CD4 activity, CD8 T cells, and it will also activate the dendritic cells. It causes the priming of dendritic cells and help the body for the upcoming attack. So toll-like receptors it will activate, and it also decreases the side effects of the glucocorticoids. Glucocorticoids, what will happen? It will cause apoptosis of T cells, which is going to be prevented by thymosin alpha 1. And in turn, this thymosin alpha 1, which will directly activate the CD34 stem cells in the bone marrow. So from that, all the CD4 and CD8 NK cells are increased. This is the figure which talks about mechanism of action, where it activates the dendritic cells and toll-like receptors, and dendritic cells are activated, which in turn, which is going to be recognized by CD8 and macrophage innate immunity is going to happen. So this is going to act both on the innate and adaptive immunity, and the CD8 T cells and B cells. B cells it will activate to Th2, which will help in the early production of antibodies for the virus and increase in natural killer cells. So this mechanism varies from mild, prophylactic, and mild, and moderate and severe. Prophylactic and mild, it will be mostly the immunostimulant, and moderate and severe, it is mostly the immunodepressant and improvement of T cells. In the prophylactic and mild, it will act to toll-like receptors and priming of dendritic cells and increase in NK cells and CD8 T cells. So the upregulation of MHC class 1 and class 2 happens here, and the class 1 which will attach to the CD8 T cells, which is already increased by thymosin alpha 1, which will attack the virus infected cells and will decrease the viral load. And the dendritic cells also activated, it activates the dendritic cells, and also it increases the concentration of dendritic cells. Both the CDC and the PDC, that is the conventional dendritic cells and the plasmacytoid dendritic cells, are activated, and there's increased production of antibody through Th2. In the moderate, it has a different mechanism of action. Since in the moderate and severe, there is exhaustion of T cells, there is increase in CD4 and CD8 cells. So it removes this exhaustion, and it will prevent the exhaustion of T cells. And it even causes the balances of Th1 and Th2, and also it causes the protection of T cells from exo activation. One, it prevents the exhaustion. Next is, and corrects the lymphocytopenia. Next, it protects the T cells from exo activation. So since Th1 is the one which causes the cytokine storm, that is prevented by the thymosin alpha 1. Next is, it activates the indolamine 2,3 dioxinase. This is the enzyme which dampens the innate immunity, and the increase in T regulatory cells, which causes the immune homeostasis. So this is the one cells called the T regulatory cells, also called as regulator of regulators, which causes the immune homeostasis, which we'll talk. It has a 3 different mechanisms. It will act on CTLA4, LA3, it will act, and it will bring the immune homeostasis. And again, it will increase the NK cells, since NK cells are decreased in severe COVID. So this is in general mechanism of action through by increasing the MHC class 1 through activating of CD8 cells. Initially, it will decrease the lymphocyte count, decrease the viral count. Then through dendritic cells activating the innate immunity. Then through the precursor stem cells, it will decrease the, it will increase the B cells and CD4 T cells. And through the immunodampening effect is through the FOXP3 regulatory T cells, which is also called the which causes immune homeostasis and tissue homeostasis. This is the molecular mechanism of action of thymosin alpha 1. And coming to the pharmacokinetics, it has a half-life of 2 hours, and after 24 hours, it will come to the baseline. It is excreted mostly through the urine. It is 30 to 60% is excreted through urine. Coming to the safety profile of this drug has a safety. This drug is available from past 4 decades, and this has been approved by US FDA and 35 different countries, and this is tried in many patients. And if you check in the PubMed, there are more than 2,000 published articles in this thymosin alpha 1. If you check the Google Scholar, there are more than 16,000 articles. And this drug has been used for more than 12 months to 18 months continuously for hepatitis B infection without any significant side effects. Even in fact, if you check the safety profile of this drug, is one of the safest immunomodulator available, because even up to 800 times the dose was increased and nothing happened in the animal studies. Coming to the side effects, it's well tolerated. It can be given for even for liver diseases, renal diseases, and hemodialysis. And the side effects is injection related side effects is the one side effect which is with all injections. And overdose, there is no reported or overdose. Contraindications, only the allergic contraindications are there. And mutagenicity, it's not mutagenic, and no major drug interactions. Pregnancy, you have to check the risk benefit ratio before giving, and nursing mothers, no data is available as such. This is a drug which was used in SARS outbreak, which I told before, in 2002 outbreak, and it was effective in controlling that outbreak. The SARS is the one which was version 1 of SARS-CoV-2. Almost the genetic is almost 80% similar to the SARS-CoV-2, and it was even as used as a prophylaxis and also prevention during that condition. So how does this thymosin alpha 1 reduces the cytokine storm? I already talked about. One is it antagonizes the activation induced anti-CD3, that is the glucocorticoid, which causes apoptosis of T cells, which is prevented by the thymosin alpha. And next is the immunodampening effect, which already told, through T regulatory cells and this enzyme indolamine 2,3 deoxygenase activity, which causes the decreased response, dampens the immune response. So this is the T regulatory cells, which is not much talked about. T regulatory cells, what happens is it is moved to the lungs. It is more, and T regulatory cells are decreased in the COVID, severe COVID. So I think there is a less of time, so I'll just move into. I'll just talk about the 3 functions here. What happens is the T regulatory cells, which will cause the antigen presenting cells in T cells through B7. So CTLA4 it will act, and it will cause the decreased activation of cytokines. So this is one of the action it has. So just a quick run through this one because of lack of time. Direct viral action is because this has been found in directly in hepatitis and HIV, and it has been found that in the in vitro studies, this thymosin alpha 1 acts to post-translational step and decrease the viral load for the hepatitis B, for the hepatitis virus and the parainfluenza virus. It has been found. These are various publications which has been published of thymosin alpha. The one major publication is the one thymosin alpha 1 reduces the mortality of severe COVID-19 by restoration of lymphocytopenia and reversion of exhausted T cells, which is published on 76 patients. So based on this study only, we have designed our 3 trials, which is ongoing. And the one more trial which has been done is the thymosin alpha 1 protected T cells from ex vivo activation. This is the one more trial which has been completed, and it has been found that they have collected the T cells from the severe COVID patients, and it has been found that thymosin alpha 1 receptors has been found in those T cells. And when the thymosin alpha 1 is added, this exhaustion markers was decreased, and the lymphocytes came to the normal. So I'll just touch upon this summary of clinical trials. So these are the total 8 trials of thymosin alpha 1. these three trials are ongoing trials in US. One is the prophylactic, one is the therapeutic, and two trials, the rest of the trials are completed. The first trial is the important trial which has shown the mortality reduction, where the drug, where the non-treatment arm with the treatment arm, there was a 20% absolute reduction of mortality, which has been found on the 76 patients. Then the next trial is a thymosin alpha 1 protected T cell from excessive activation in severe COVID. So that's I've already told about where they collected the blood from the severe COVID and found that the thymosin alpha receptors was found in their T cells, and this thymosin alpha was added, and it has been found that the exhaustion markers were decreased. And the one more trial is the prophylactic trial, which has been done, which is on the 3,000 healthcare workers, where the thymosin alpha 1 and interferon alpha nasal drops was given, and none of the healthcare workers became positive. That means at that time there was no RT-PCR, but the endpoint was respiratory symptoms and chest X-ray. So all the and it was controlled with the literature arm, where it was found almost 2,000 healthcare workers became positive. I think because of lack of time, I'm going to stop here. So there are trials which are there, so which I'm not going to touch upon. I think I'm handing over the session to Dr. Wadhwa.
Thank you, Dr. Adarsh Shetty, for the wonderful presentation. With this, I'd like to request Diana to play the pre-recorded session of Dr. Rahul Pandit. We'll be there in a couple of minutes while the session is on to address our concerns as well. So, Diana, just can you play the recorded version of Dr. Rahul Pandit?
Yes, sir.
Thank you. And Diana, just keep it on unmute. Your audio is muted.
ऑडिओ नाही शेअर केलं तुम्ही
केलं
आता केलं
नाही तेव्हाच केलं
मग आवाज नाही येते
हा
And we decided that to have the proof of concept. Good evening, my friends. It is lovely to be here for this evening talking about this molecule, thymosin alpha 1. I am Dr. Rahul Pandit. I'm the Director of Critical Care at Fortis Hospital, Mulund. And we decided that to have the proof of concept, we should probably do an investigator-initiated clinical study. So here I am presenting you the protocol for a prospective single center in investigator-initiated clinical study, which was done essentially to evaluate the efficacy, that is effectiveness, and safety of thymosin alpha 1 in severe COVID-19 patients. So we chose to basically use only the severe category of patients, which means the PF ratio was really poor and they were all intensive care patients. Now, after listening to this fantastic talk from Dr. Shetty, you probably know now that it is this T cells which get exhausted and the lymphopenia, which is one of the bigger problems of COVID-19 patients. And to help modulate that immune response is where the thymosin alpha steps in. Now, is this something which is new? Has this not been tested before? That's not the case. If you look at this study, and if you look at the principle in which basically COVID-19 affects the body, you will see that the COVID-19 infection primarily has basically affected the pulmonary function by fluid accumulation and increased levels of immune cytokines that can damage normal tissue. Viruses further compound this reaction by converting the host cell to replicate the viron, amplifying the immune response. So essentially, there are two things which are happening. One, that the virus is causing a problem, and then the second thing, the immune response, which gets amplified because of the viron. Obviously, the first stage and the first step of medication, whenever we talk about any immune response, has been corticosteroids. But we all know that corticosteroids also have a double-edged sword and can cause immune suppression, as well as from our H1N1 data, we know that it can cause a delayed viral clearance as well. Hence, thymosin alpha 1, which is a polypeptide hormone produced by the thymic epithelial cells, can effectively increase these exhausted T cells, support its differentiation and maturation, and most importantly, reduce the cellular apoptosis, and thereby decrease the lymphopenia and have the lymphocytes mature. The clinical data for this started off with the 76 critically ill patients of COVID-19 who were treated and admitted at the general hospital in the command hospital of Wuhan Pulmonary Hospital. And this study happened between the December 19 to March 2020, which provided us the initial data of thymosin alpha 1 having benefits when used for treatment of COVID-19 patients. What it showed was that it essentially reduced mortality as compared to the patients who were basically not treated with thymosin alpha. And thus came the proof of concept that it looks like a potential therapy which can be used in combination with the standard of care with COVID-19 patients. Hence, we planned this study to evaluate and compare the efficacy of thymosin alpha and its safety as well. The primary objective, as I've always said, was to look at the efficacy of thymosin alpha 1 in COVID-19 patients. And the secondary objective was to see if it was associated with the clinical response and to evaluate the safety in severe COVID-19 patients. So this is how we sort of came out with the primary objective that this is what we're going to do. The endpoints was duration of ICU stay from baseline to the end of study day, which is day 7. So it was a 7-day treatment plan. Ventilator duration, incidence of all-cause mortality. Time frame was from the date of drug administered, the date of hospital discharge, or date of death from any cause, whichever may be first. And the patients were followed up to 28 days. Duration of hospitalization obviously was also one of the primary endpoints which we were looking at. The secondary endpoints was change in the total lymphocyte count, CD4 and CD8 count from the baseline. We also looked at some of the other cytokines which are known to increase along with the COVID-19. And that's a part of the immune response which happens, which is the ferritin, IL-6, LDH, CRP, D-dimer. So we did a baseline value for this and then we followed them up to see what changed in them. The oxygen saturation, increase or decrease from the baseline. And we looked at the clinical progression based on the 8-point ordinal scale on day 1 screening and at the end of the day 7 study day. We also looked at the number of treatment emergent adverse effects and serious adverse effects which happened during this study. The study design essentially was quite straightforward. There was a screening visit, then there was an enrollment visit and a baseline visit, all of which usually happened within the first day. Obviously, hospitalization from day 1 to day 7 as per investigators' discretion. But here, it was not hospitalization, it was intensive care admission. in severe COVID-19 patients. End of the treatment or end of the study was 7 days, and follow-up was up to 28 days. If the patients were out of hospital by 28 days, a telephonic follow-up was done. The total study population was basically based on following criteria. We had some inclusion criteria, exclusion criteria. We took into the account some patients may withdraw from therapy, number of subject, and study duration. So, as I said, the treatment was for 7 days and follow-up was for 28 days. To have a proof of concept that it is efficacy based as well as safety based study, we decided to use 15 eligible patients who would be enrolled after a proper consenting process. And we went through the vigors of the ethics committee. The ethics committee approval was sorted, as well as the CTRI registration was done for this trial. I can tell you that we have finished the inclusion and we finished the recruitment for this study, and the data will be been analyzed right now. Obviously, it takes some time for the data to be analyzed to be presented before you. The inclusion criteria were male or female greater than 18 years of the age at the time of consenting, patients who could and were willing to provide written informed consent, severe acute respiratory syndrome coronavirus 2 infection confirmed by PCR test or any other confirmatory test as mandated by ICMR in India. If the patient presents any one of the following features: respiratory distress with respiratory greater than 30, SpO2 less than 90% on room air, PaO2 less than 200 millimeters of mercury, or a patient representing respiratory failures and required mechanical ventilation. The fifth criteria was that if the patient was not able to give a consent, which in intensive care unit it happens quite often because the patients are quite sick, the legal next of kin was requested to give a consent, and he was willing to participate in the clinical study and basically was able to comply with the protocol. Exclusion criteria was if the patient had participated in any other clinical trial. Fortunately, we didn't have any clinical trial running at that period of time in our unit. Patients with presence of any pre-existing illness that we thought would put the patient in any unreasonable risk, like a malignancy or anything. Patient who had participated in another trial with an investigation drug within 1 month prior to this trial, and patient who, in the judgment of the investigator, is probably unlikely to comply with the requirements of this protocol, we decided to exclude these patients. The study schedule was basically based on these around 14, 15 parameters. And as you can see, the study visits were day 1, day 2, day 6, and day 7. And what did we look at? What were the various things, various variables we looked at? We looked at the hospitalization, consent, demography, medical history, physical examination. The first 4 were on the study day. Then every visit had a physical exam and the vital signs, chest X-ray, clinical lab test, biomarker assessment was done on day 1 and then on day 7. Obviously, the clinical progression and deterioration was looked on day 1 and day 7. Oxygen requirement was looked at all the other visits. SpO2 was looked at all other visits. Respiratory symptom assessment were done. Study drug administration was looked at all visits. Concomitant medications, and the most important thing, adverse effects. We looked at adverse effects at each part of the study, and fortunately or unfortunately, that was the most important thing which we wanted to actually look at, whether the safety of the drug was good or not. Now, what was the study medication? After the baseline assessment, all patients were administered 2 subcutaneous injections of 1.6 milligrams of thymosin alpha 3 times a day, along with standard of care for 7 days. Standard of care medications were as per the guidelines of Government of India, Ministry of Health and Family Welfare, and the task force of Maharashtra government, which essentially had almost similar guidelines for managing these patients. The thymosin alpha 1 comes in a lyophilized powder. It is reconstituted with 1 ml to provide the effective solution. And after reconstitution, the final concentration of thymosin alpha was 1.6 milligram per ml. So that means that 3.2 milligrams were given for these patients. The two subcutaneous doses were basically administered. The drug was stored in the 2 to 8 degrees centigrade in our pharmacy. And once the two subsequent injections were started, they went on for at least 7 consecutive days, which means that every day the patient got 9.6 milligrams per day. Risk benefit assessment. Thymosin alpha 1 had benefits of COVID-19 patients, especially with severe lymphocytopenia, and had also reduced the mortality of COVID-19 patients compared to those in the untreated group. And in the treatment arm, a summary table with subject compliant to the patient and non-compliant to the study was going to be provided. And patients were basically who took around 80% of the study drug considered to be compliant. Now, adverse effect. We know that thymosin alpha is a drug which has been there for the last 40 years. It's well tolerated. Even in patients with decompensated liver failure, renal disease, requiring dialysis, patients with primary immunodeficiency. The only problem is that it is a subcutaneous injection. So there is the injection site redness and tenderness which can happen. And there are no clinically attributed side effects of thymosin alpha, which are basically reported out. These are some of the references from the previous studies. As I said, the two studies from China were one of the more important ones. And then there are some other studies which have looked at some other causes for thymosin alpha. And essentially, we are hoping that once the study gets analyzed, we will have some concrete results to present to you. So this is how the study was planned. We've completed recruiting the last 15th patient last week. The patient is also coming up to the 7th day of his follow-up, and then would be followed up 28 days. We will have a few more days, a few more weeks before we actually complete our complete analysis and follow-up, and then the study would be basically deciphered for us to understand how did it fare. Thank you very much. I'll be happy to take questions after this. Thank you.
Thank you, Dr. Rahul Pandit, and thank you, Diana, for this video. Sir is already there with us. We would like to have a few comments from Dr. Rahul Pandit before we go on to the case presentation from Dr. Amit and Dr. Joshi. So, sir, over to you for your valuable insights and comments on your experiences about this protocol. But natural, because of ethical reasons, we cannot share the results as of now because these are under analysis. But definitely, yes, about the rationale, the scientific justification for this as a summary, which would be important for the audience to learn, sir. Thank you.
Yeah, no, of course, I think no doubt about that. We've finished that study recruiting. As I said, the last patient is just finished at day 7. So we've got a few more weeks before we can actually go ahead and talk about the results. The whole concept was to try it out in critical ill patients, which was the main important thing we did. We tried to keep the protocol as simple as possible so that the critical ill patients could be sort of identified based on their hypoxia and the oxygen requirement. And we found that some of the simple observation was that it was not a very difficult protocol to follow. The regime is quite reasonable to follow. The nurses did not complain about it. It was too labor intensive for them to give this injection 3 times a day. And some of the patients, when they were recovering, also did not complain about the pain in the abdomen that much, which is what my main concern was. So these were the simple things which I could talk about. There would be questions coming up from audience. I would be happy to take up a few questions when they come up. But I think what prompted us to do this study was that it makes a lot of physiological cells to improve your lymphocyte counts. And if you have a drug which is able to sort of immunomodulate from that direction, but not have the immunity knocked down, which the other immunomodulators which we are currently using is basically doing, which is the steroid as well as the MABs, then that made a little bit more physiological sense and a safety sense for us. However, as I said, this 15 patient study is not going to be the game changer study which will actually make it, but it will further prompt up some bigger studies to actually help us understand whether this evidence is going to be a robust level 1 evidence, or we are going to have this as one of the important armamentariums for treating COVID, but along with the other standard of care. So that sort of remains to be seen, but it is certainly a step forward in the right direction.
Very well, sir. Thank you so much for your comment. I think that's a very important, vital thought that it's compounding with the ongoing therapies. In fact, that's the differentiated approach, I would say, for that matter, that we don't have any specific therapy for this particular disease per se. But I think one element which is strongly being implemented is that the rigorous way of approaching this molecule in terms of this specific disease. I think a lot of questions also have emerged in that direction. We can take the questions at the end after we finish presentation from Dr. Amit Gupta and Dr. Vidoshi, because anecdotal experiences translating into small case series, case reports, and will also address some of the specific questions what we are encountering during the chat. So without wasting any time, I would request Dr. Amit Gupta, he's from Calcutta, to share his experiences as case reports in this particular scenario. Over to you, sir, Dr. Amit Gupta, please.
Okay, I guess my slides are visible to all of you, and
Yes.
I'm audible.
Yes.
Yeah. You're audible. Your slide is visible.
Okay, fine. So I know in this pandemic time we are all busy, so I'll straight away get into my case. At the start of pandemic, we were quite focused on the etiology. We knew we need to kill the virus. Hydroxychloroquine, remdesivir, favipiravir, no response. Later on, we thought we need to boost the immunity. So zinc, vitamin C, high dose of vitamin C, vitamin D, still we did not get any response. Then we decided to reduce the immunity. Steroids, high dose of steroids, IL-6 inhibitors, tocilizumab, still no response. Where do we stand? What do we do in such a situation? The answer that now we are sorting to is to regulate the immunity. And how do we regulate the immunity? We regulate the immunity by introducing something which is very new, and that is to introduce the component of intelligence in immunity. And what does such introduction of immunity with intelligence happen? It means something that will stimulate the immunity when the immunity is low, but would also take care to dampen the immunity when it is overstimulated. So striking this balance is actually the need of the hour. And based on that, I am presenting a very simple, very practical case, which most of us are going across during this pandemic. So this is one of my old patient. He is a 73-year-old man. I would rather add here a stubborn old man who presents with fever, cough to the GP and found to be COVID positive. His entire family was gradually getting COVID positive. And everyone knew that he would not get admitted because he has got a hospital phobia. So he was started on with the etiological treatment of favipiravir. He was a COPD patient, so a steroid. He was given enoxaparin and antibiotics at home. So he was pretty much on a very regular, typical mild ICMR guideline covered patient. The comorbidity that he had was diabetic, COPD, and hypertension, along with elderly group. So he had quite a long list of comorbidity. On day 3, his condition started to deteriorate, which is such a common thing you all know. So saturation went down below 94, which is a typical criteria where we advise patient to get hospitalized, to which he denied. He refused admission. The family member waited till day 7, where his condition deteriorated further. He was put on oxygen at home. They were trying to give a prone position to get some result, but that was failing. With that panic situation, I got a phone call from their family relatives, and then they forced kind of a situation, decided to admit the patient. His condition worsened, and the patient was admitted to the hospital with a saturation of only 86 in room air. Generally, patients who get admitted in our unit, we try to prognosticate. Along with the history, we do 1 or 2 extra tests to give family relative a prognostication. On that, we try to do a cortisol, which, if high, falls into a bad prognostic criteria. And we also do, especially in elderly male patient, testosterone, which suggests in case of low testosterone, a higher risk of ICCU conversion and a poor prognosis as per all the recent trials. So he had a pretty normal cortisol response. It was not high, but testosterone was quite low, which is suggestive that he may not have a good outcome. Even before going to the bed, we try to do a CT scan of patient, and these were the lung lesions. You can understand from these parenchymal ground glass opacities that it was getting progressive in nature. And we later on tried to exclude any possibility of pulmonary embolism because he's a long-standing COPD patient and was suffering from COVID, generally non-ambulatory kind of a lifestyle. So this was the typical scenario with which the patient was received. Upon admission, these were the prompt blood reports that we got. So you can see from here the highlighted ones, WBC, which was above 15,000. He had a very low lymphocyte. Please note that, and the ratio of neutrophil is to lymphocyte, which was also quite high. And we know, after hearing so many episodes of these lectures, that these ratios, these values are actually important in the outcome of such patients. To add further to it, we have a high CRP. Our normal cutoff is 0.5. So you see a CRP more than 10 times here, a rising ferritin as well. These are the blood reports, and I have already shown the primary ones ahead. This was on day 2 of admission, and now we have the reports available for interleukin 6, which was sent yesterday, day before, and you can see over here there is a rising interleukin 6. So it was going up. Patient also started to lose its blood sugar control. The same medication with which patient was getting controlled on his blood sugar has now started to get uncontrolled. Probably it was the insulin resistant effect of COVID, or probably because of the concomitant steroids. The oxygen saturation started to again desaturate, reduce. Now we have 89 with only 3 liters, and gradually the oxygen requirement was going up. By that time, we got the CT angio and the HRCT report as well. You can see he was a typical CORAD 5 progressing, and up to 50% of lung parenchyma that was affected. Mind you, he is a patient of COPD. We do not expect large amount of functional lung in such patients. So even a 50% lung involvement means a lot for such patients. On investigation further, we found that the lymphocytes started to improve with the introduction of Immunocin alpha, which we gave on 17th, that is on day 2 of admission. And we believe a full therapeutic dose of Immunocin alpha with 2 vials thrice daily. As you know, that this drug has got a rapid absorption of 2 hours into the system. but unfortunately a rapid excretion as well. So not a pulse dose at a time, but a breaking dose of 2 or 3 times in a day is important. You know that the T half of immunosin alpha is only 2 hours. So you expect it to work for roughly around 4 to 6 hours or maximum 8 hours. So you need to repeat for severely ill patient more number of doses as per the need of your patient's severity. So with that dosing on the 17th, you can see that the lymphocytes started to rise in number. We know by our prior speakers that lymphopenia is a marker of T cell exhaustion that we commonly see in COVID patients. So stimulation at this stage of lymphocyte is paramount important to mount a failing T cell response. And you can also note over here that there is a gradual improvement in the SpO2 with the same amount of oxygen that was maintaining 89 has now started to maintain a 94% of saturation. So we all started feeling better with the blood parameters report improving as well. The CRP, which was, if you can recall, above 5 has now gone to 4. And on the very next day, though the lymphocytes did not improve remarkably, but other parameters, especially the CRP, considerably started to fall down just in a matter of 2 or 3 days. So we started to see a clinical improvement. The dose of immunosin alpha was still the same, 2 thrice daily. And that was the only introduction that I made in this patient right on admission. Patient was already taking all the drugs that we generally have in our protocol. So I need not add much. He already had an antiviral. He already was on a good amount of steroid. He was on enoxaparin. So the only addition that was made on hospital was immunosin alpha. And with that, the changes were quite remarkable and quite encouraging. These are further follow-up reports, and you can see that the CRP had a steady fall and a good improvement of patient, with now the SpO2 rising up to 96% with only 2 liters of oxygen. So now we get requirement of oxygen going down and oxygen saturation improving. This is yet another value, and you see the CRP has gone down even further with an even improvement of the SpO2 to 98% with just 2 liters. Just note that lymphocyte has now climbed up to 15. So we now get an idea that probably the T cell exhaustion, which is common, and the interleukin 6, which was quite raised, has now started to fall as well. So the impending cytokine storm that probably we all know happens just after such a blood report was probably avoided, and patient gradually started to improve to all our expectation. These are the reports, and I have actually highlighted most of them. Just go through these reports. It's like 98%. This is the lymphocyte improvement and CRP almost touching the normal value on 24th. And immunosin alpha introduction. This is the summary of the lab parameters that I was talking. You can see the only thing that has improved over a period of time is the lymphocyte percentage, which is a good prognostic marker. Rest, all the inflammatory parameters and the need for oxygen has gone down, as you can see from those multicolored worms on the top. So this is the nutshell summary of day-wise progress of the inflammatory markers in numerics. And these were the treatment regime that finally patient was on the final day of discharge, with injections, converted to oral tablets. So that was a brief presentation of my experience with immunosin alpha. Thank you.
Thank you, Dr. Amit Gupta, for your wonderful experience sharing through the case report. With this, I like to hand over to Dr. Ravi Doshi to kindly share his experiences again through a case reports from a differentiated perspective. Thank you so much. Over to you, sir.
A very good evening. Thank you, Dr. Navneet, for giving me the opportunity, and of course, thank you, Gurfik, for letting me share my experience on this very wonderful molecule that we have come to discuss upon. And frankly, fighting with the COVID pandemic for the past since March almost, I really was thrilled the moment that I came across a molecule like Immunocin alpha, because it has solved a lot of problems which we as physicians, chest physicians, intensivists, all are put together, have been facing in the management of COVID since the beginning of the epidemic. That very problem is that of sudden deterioration. That very problem is of difficult to treat and life-threatening cytokine storm. That very problem is of the dual nature of tocilizumab, availability of tocilizumab, pricing of tocilizumab, and how immunosin alpha in itself helps us to counter the cytokine storm in a very different way, in which the same idiom that prevention is better than cure comes to mind. Now, we at Shri Aurobindo Institute of Medical Sciences at Indore had the opportunity to compile a paper of 365 patients of coronavirus disease at our center. And in that particular research that we did, we had almost 329 adult patients and 36 pediatric patients with comorbidities in 47.11% of patients. What was most important is presence of lymphopenia and comorbid conditions were identified as risk factors for the requirement of oxygen, mechanical ventilation, and death. And if I look at this in my work done from March, April, and May, from what I see when we have started using this molecule in August, September, and October, we find that to a huge extent this has helped us improve our outcomes. Now, as several prominent papers and our speakers, initially Dr. Shetty, Dr. Pandit, have highlighted the fact that high neutrophil to lymphocyte ratio is a very important prognostic marker. The risk of in-hospital mortality is 8% high for every unit increase of the neutrophil lymphocyte ratio. It is very important in clinical routine to screen the prognosis. One very interesting fact I will like to share with you, that we were part of several trials which also involved plasma therapy. So in those patients, we used to give plasma to some subset of patients. There was very high mortality. In fact, in our arms having high mortality, we found almost all of them had a very, very high neutrophil to lymphocyte ratio. So what we understood very early, somewhere around in April and May, is that if we have something to control this neutrophil lymphocyte ratio, we might actually cause some difference or dent in the mortality of COVID-19. Now, I would like to present two cases. They are very simple cases. A 70-year-old male patient presented with fever and shortness of breath. The patient also had hypertension, type 2 diabetes mellitus, and chronic kidney disease. At the time of admission itself, oxygen support was required. We initiated a treatment regimen of meropenem, clindamycin, steroids. We started immunosin in the highest dose possible of 3.2 molecule, 3.2 milligram thrice daily subcutaneously, along with low molecular weight heparin. This patient initially on admission had very high markers, a CRP of 2.4, LDH of 264, D-dimer 2834, interleukin 6 was very high, 105, ferritin was 938. The neutrophil lymphocyte ratio was 6 at the time of presentation. Over the period of time with treatment, we found that the lymphocyte ratio came, the neutrophil lymphocyte ratio came down to 15, and eventually the CRP came to 0.6. This very high interleukin 6 level told us that the patient with admission in itself was in cytokine storm, but due to some issues regarding availability, we were not able to administer any other agent. we decided to take up Immunocin alpha, and we used this patient, this particular molecule to very good effect. And from a situation where on day one, the patient was in cytokine storm, without giving any other anti-interleukin 6 or other antibody, we were able to counter the storm and take out the patient successfully. Despite of this diabetes, he on CT, he had around 15% lung parenchymal involvement on the last day, the 29th and 30th. Initially, because of the oxygen requirement and his financial condition, it was not possible for him to undergo higher radiological investigation. The patient's family was more interested in treatment rather than investigation. So at the end of the treatment, when they saw good improvement, we were able to get the CT done. X-ray initially showed bilateral diffuse involvement, but at the end, we were happy to see that not only the cytokine markers came back to normal, but even his creatinine started normalizing at the end of the treatment. The CRP final was less than 0.6. Now, I would like to present one more case of a young male. This case is very interesting. Why? Because it is a readmission. A 39-year-old male patient presented with fever, cough, and shortness of breath. He tested positive on 11th of September. The chest CT showed 35 to 45% lung parenchymal involvement. He was started on remdesivir, antibiotics, steroids, and antithrombotics. Oxygen support was given. He clinically improved. So he was discharged on the 17th, but the very next day he had to be readmitted with shortness of breath, falling oxygen saturation, and from an initial 35 to 45%, the chest CT showed an increase in the lesions of 65 to 70%, despite the patient receiving a full course of remdesivir. So this time we started him on immunomodulator Immunocin alpha, and we gave oxygen support, steroids, and antithrombotics, and we were successfully able to discharge the patient on 20th. After 10 days, we gave 7 days of Immunocin alpha therapy to this particular patient. Now, at the time of first admission, the patient's ferritin was 453, but at the time of second admission, his ferritin levels were high, and his lymphocyte ratios, his lymphocyte was 13. The neutrophil lymphocyte ratios in both cases was 8 and 6 respectively. Now, one very important part which we came to understand, that this particular time using Immunocin alpha was a bit challenging for us. But as and as we gave Immunocin alpha to this patient, and as and as his days of readmission started, we could see clinical stability coming, not only in the reports but also clinically. And the patient, in fact, by the day 3, commented that my recovery is much better than my first admission. And by day 3 of the secondary admission, we were able to take off his oxygen. And despite this 65 to 70% of involvement, with immunomodulator therapy, which we instituted on the second time, we were able to achieve a better symptomatic control, surprisingly, in this case. And finally, the patient was discharged successfully with all his inflammatory markers coming negative. So these are two short cases. There are many more. There are many scenarios where Immunocin alpha has taught us that it can be used beautifully, particularly chronic kidney diseases. And we even had some cases whereby some patients could not be given steroids because of one very uncontrolled sugars. In them also, Immunocin alpha showed a very beautiful role and helped them to prevent cytokine storm. In fact, one very interesting observation by my pharmacist was, after we started using Immunocin alpha, our anti-interleukin 6 antibody, there were a good 12 days where we did not need to use our IL-6 antibody even once. Thank you very much.
Thank you, Dr. Doshi, for the excellent deliberation on the specifics of this case, and a very distinctive case. I think we can take quick 1 or 2 questions, because so many points are already covered up during the discussion, which are already there in the question format. I'd like to address one question from Dr. Devendra Mida from Mohali. He has categorically asked with the perspective of the impact and the impact on the total lymphocyte count, CD4 and the CD8 count, with respect to ratio of CD4 and CD8 plus. I think this is already being addressed marvelously in the cases. I think this is one area where it's a direct implication which are beneficial as far as the measurable markers are concerned. I'd like to ask one question to Dr. Rahul Pandit that uh, what about cytokine storm as far as when the patients are already on thymosin alpha on this molecule?
Yeah, look, I think the so if you basically care for these patients properly, and you have an immunomodulator which basically reverses your neutrophil lymphocyte ratio, starts improving your lymphocyte. Technically, you should not have the need to get into a cytokine storm for a patient at all. That is the basis for that. You would probably be able to avert it quite nicely. I don't want to divulge anything right now more than that, because as I said, I would like to keep the sanctity of the study there.
Yeah.
So we feel that technically it is the one thing which will actually avoid the cytokine storm. And one of the two studies which were presented actually showed that they did not go into cytokine storm when they were actually thought to be going into the cytokine storm. So I think we need to basically be very clear that when the study comes out, we will be able to publish that and we'll be able to talk about the details. There are 2-3 questions here. Prashant Kumar has asked actually that if so many patients in Mumbai, why didn't we include more? If you remember, Dr. Kumar, that our main reason why we did the trial on 15 patients, study on 15 patients, was we needed to look at the safety and efficacy both. So this is a proof of a concept, and I think if it works well, we will be able to look at several larger patients across Mumbai, across India as well. So this was one of the things which I thought I should answer out.
Right. And also from a regulatory perspective, one of the purpose is to have a regulatory perspective in place, which actually defines the threshold of the number of patients in that regard. I think we can take up all the questions because we always have we are a listening organization in that regard. And every Wednesday 9:30 p.m. we have a live session for 1 hour to address all the question answers. So the paucity of time. And I would like to, at this juncture, I would also like to thank Dr. Rahul Pandit, Dr. Adar Shetty, Dr. Amit Gupta, and Dr. Ravi Doshi for their efforts and excellence for bringing the science of Immunocin alpha at the forefront in that regard. With this, I'd like to hand over the session to Samrat and take it forward. Over to you, Samrat.
Yeah, thank you, Dr. Wadhwa. And with this, I'm we are on the verge of closing the entire thing. So just my closing remarks on the same. Yeah, this webinar was sponsored in technical collaboration of Gufic Biosciences Limited. For any further queries, you can also write to us at info@guficbio.com, where our medical team can get to you back with the response. And also, as said by Dr. Navneet Wadhwa, every Wednesday from 9:30 p.m. to till 10:30 p.m., we have kept exclusively for answering the queries of you regarding Immunocin alpha. And our medical body will be there, our medical doctors will be there to answer your queries regarding the molecule. And this webinar was sponsored by Immunocin alpha, lipidase thymosin alpha 1, which modulates immunity perfectly. And with this, I am handing over the session to our Assistant Vice President, Mr. Shekhar Ale, to provide his vote of thanks.
Yeah, thank you, Shamrat. Good evening to all. Regards from Gufic Biosciences Limited. It's our great pleasure to thank all delegates for joining with us from India and Nepal. We are thankful to Honorable Speaker Dr. Rahul Pandit, ICU Director, Fortis Hospital, Mulund, Mumbai, who has given his valuable time for investigator-initiated trial on severe COVID-19 patient with Immunocin alpha and sharing his experience. As per ethics protocol, He has already told that he will be able to share the details of the trial after completion of all the regulators. And after completion, that will be unveiled very soon to all through a webinar very soon. We are thankful to Dr. Amit Gupta from Kolkata and Dr. Ravi Doshi from Indore for giving their valuable time to share their experience through case presentation. We are thankful to Dr. Adarsh Sethi for his presentation on the role and benefits of Immunocin Alpha for the COVID-19 patients. We are thankful to Dr. Navneet for all coordination and medical supports. As the flag bearer, Gufic Biosciences has taken initiative to update medical fraternity regarding the role of Thymosin Alpha patients' outcome through series of webinars in various stages of COVID-19 patients. So far, we have completed 3 webinars and with this one, and on 30th October, we will have the next webinar, which will be led by Dr. Om Srivastav, the leading ID specialist from Mumbai. Along with him, there will be 2 doctors from different cities with their case studies. I'd like to take the opportunity to invite you all to the next webinar. However, our team, our colleagues, again, will invite you individually. Within few months of the launch of Immunocin Alpha, the response are very, very encouraging from across the country. We are getting daily positive feedbacks in terms of patients' response and COVID patients. So far, more than 1 lakh injections have been used across the country. We are thankful to all those who have used, who have passed the benefits of Immunocin Alpha to COVID-19 patients. We are sincerely thankful to all doctors for passing the benefits of Gufic's world-class quality unit lifeless products to millions of patients. Once again, thank you all for joining with us. See you again in the next webinar. Good night to all. Thank you, sir.
Thank you very much. Thank you.
Thank you. Thank you.